begin just like run through your career, going like from your undergraduate years to your position now.
Okay, so as an undergraduate at Penn State, I was what would be called a late bloomer for the times, and a very late bloomer by current times. Most of my colleagues knew from an early age they wanted to be scientists, and I found science very boring when I was younger, and just because of my own ignorance in education. So, but I was very influenced by a particular bio professor who just seemed to have a D life. He taught a class that was kind of fun, and he seemed to love life, even though he was chronologically old. He got to hang out with kids and teach, and I thought being a professor meant teaching, and so when I got my degree, I was like, shit, I got this bachelor's in science, because of my first year I thought maybe I wanted to be an MD or something, because that would make your mother like you more, and maybe you'd get chicks. And so I realized I'm averse to touching strangers or poking them with blood. I don't like blood, so I wouldn't be a very good doctor. Here you are with this bio degree, good GPA, and someone was willing to pay me to go to grad school. I have to tell you that that is really bad to admit. I was closeted about this for a long time, because you're competing against people who've known their whole life. And I don't understand how a 16-year-old knows these people doing research, that this is what they want to do when they're 20. And I was just very lucky. I had no idea what grad school, or even really being a scientist, meant, that I snuck into grad school. And despite my lack of experience, I caught up and then fell in love with what I accidentally stumbled into. And so the sad testament is that by today's standards, I would not let me in my lab or in our grad program. But I think it's normal to not know what the hell you want to do until you're 21, 22, 23. That's the honest story. The background story is I had a little bit of a lab gig in one lab where I washed some dishes, but it wasn't really science.
Oh. And then how did you get your position here at UT?
At UT?
Yeah.
Well, there's been a lot since then. So did my undergrad at Penn State, then did my graduate work at Pitt. Wanted to do cancer research, so I thought, again, that's a very common rookie thing, that you want to do something incredibly translational and health relevant, because you know, that's what everyone would think until you get in the trenches and appreciate the beauty of basic science and the glory of serendipity and being the first to conquer a problem and the unforeseen benefits to humankind. But when you're starting off, you're just like, I want to cure cancer. Yeah. The only relevant cancer lab in my program was a cancer virus lab, and that's how I became a virologist by accident. I wanted to be a cancer biologist. I fell in love with my thesis advisor. He was badass. He's a mix of like Jerry Garcia and John Wayne. He's like the perfect spitting image. That's actually him there on Virus Hunter magazine in Siberia with his horse and rifle riding through the plains looking for arboviruses.
It's a common thing with virologists, just being super cool.
No. It's 50-50 like all scientists. Some of the most interesting people you'll ever meet are scientists, and some of the most highly annoying, socially challenged are also professorial monologuing scientists. We get good at monologuing, you gotta kill time, you know, while you're teaching. So then I was a virologist by training, in a small, puny virus that was incredibly important in its heyday as a model for cancer. It causes cancer in the right model systems, and cancer is really complex. You know, lots of factors cause cancer. In this small little virus, you could put it in. It was only 5 little kb, maybe 7 genes or less. That was enough to cause cancer in the right system. So it was a reductionist approach. But in that process, I fell in love with viruses. I felt like if you're ever going to have any absolute truth in your system, the less genes, the less complexity, the better. But there's a problem. If you study a virus that's puny with only 7 or 8 genes, it's an incredibly competitive, cutthroat, ruthless thing. Science is beautiful when done right. It's collaborative. We stand on the shoulders of our predecessors. But you want to cut the heart out of your competition. And it's ruthless when there's only 8 genes and there's 25 labs on that virus. So I wanted to get us to a situation where there was a tumor virus with more open reading frames and there were labs studying it. And that led me, and it had to be in a cool city, so that led me to San Francisco, UCSF, working with one of the world's most renowned virologists on a bigger tumor virus. And from there, after that, got a little luck, made some discoveries, peaked at the right time. began a little late. It took me 5 years of postdoc. Maybe the average these days is 5, back then it was probably 4. And then we were a bit of a commodity, and we could have worked at several places. To be honest, shouldn't admit this, but Austin was supposed to be my practice interview. It was my first place I interviewed, and I forced them to be my first because I wanted to get battle hardened before the places I was really going to get my job at. And when I came here, I don't know how to explain it, it just felt like home for the first time in a long time. I mean, I love San Francisco, I love Pittsburgh, but it didn't feel like home. And I can't explain why that was exactly. One of my colleagues is so weird and out there that I felt like no one would think I was weird. He also told me the reason he picked Austin was, well, he was actually harassing me because there was like a bit of a competition. Several places wanted to hire us, and I was trying to make the decision, and he would kind of prank call me and be like, "You want to come to Austin because the air smells better," and even hang up. And so like the two things that stuck with me is like, he's fucking crazy, which I liked, and then I think he's right. I mean, I thought it was a metaphor, but when I finally came here, he was like, no, I was being literal. The air smells better. So that's kind of how I ended up here. And it's been a good choice. So I didn't want to, I'd spent 5 years at a hospital setting. You walk past literally people in their most miserable states on your way to work, and you really want to help people, but it kind of sucks compared to being around this. So my main job is to do research that brings money into the university and maybe changes the world a little bit. But the part of that here, unlike the other places, is we get to teach and hang out and forget how chronologically old we are. And so UT was the place that had the best, overall combo of really good science, but it didn't have to be clinically relevant. We just bash tumors and look at genes that are different. We do, you can do anything you want in any area, and I'm just not good enough to pick where I'm going to go, but I am good enough to know when we're lucky, and so we can traverse all kinds of things. And UT is really strong in big biology, systematic biology, large data, things that at the time were not applied to traditional virology. And so there was something I could create here, I thought, that couldn't do. I could have been a plain Jane virologist anywhere, I suppose, but here there were options to maybe do something different, dance to our own tune, or however you want to put it. And that's the story. Here we are.
No point you should touch any sick people.
I probably touch a lot of you guys every time I grade a paper or something. But I have a theory that when I'm older, I'm going to be better off. So people who contact young people who are just like really bags of disease, whether it be children or college kids or whatever, you get a little bit of a vaccine effect that I won't get that particular rhinovirus ever again. So maybe when I'm older. So your immune system peaks functionality-wise at age 12. So the ability to generate an adaptive immune response, functionality like responding to a particular antigen, peaks at 12. But you get less and less colds over the years to a point because you get memory, immune memory. So I feel like while my immune system still works, maybe when I'm 70, I'll have an advantage for hanging out
over all the other professors.
Okay, so I was reading one of your papers, and it was kind of hard to understand because of the technical jargon, but one of the key words was microRNA, so could you please explain what that is and its role?
Yeah, sure. MicroRNA was a major influence to my career, and it was, it is a huge part of what our lab's interested in, but not the only thing. This started maybe, about 15 years ago. There was a bit of a mini revolution in molecular biology, maybe the biggest thing that happened in the previous 20 years. It was called RNA interference, and it was the fastest Nobel Prize from the time of publication until the prize was awarded in history, one of the fastest, and this was to Andy Fire and Craig Mello. What Fire had discovered was complete serendipity, just total dumb-ass luck, where he was studying worm development in a nematode, and in the process of studying one of the genes that was involved in larval staging, he did an experiment where he put in what was supposed to block the gene. It's called antisense, so a piece of RNA complementary to the mRNA should block its function. he did that, and he got a really cool phenotype, exactly consistent with his model that gene was involved in biology. Many people would have taken that to the bank and got their big publication, but he is he's an honest scientist, and honestly, science is muddy and dirty and never is clean cut, and almost certainly your ideas are almost always wrong. So he did the control, didn't have to do this control, but by luck, he did the control where he put in a small piece of the gene in the sense orientation as well, which shouldn't block it at all, right? Because it's not complementary. And it blocked it just as well as the antisense. And so maybe people would have swept that, other people would have swept it under the rug. He pursued it, and he had the breadth of knowledge to know that when you in vitro transcribe RNA, a small piece of it, a small amount of it, has false priming from the bacteriophage polymerase. And so even though you're putting in 99%, let's say, about antisense or sense into that worm, you're putting in about less than 1%, but still, of double-stranded RNA. And so that was the key. And he knew that about the phage polymerase. And so his dumb luck choice of the control and his honesty not to ignore it led to the fastest Nobel Prize from publication. This is called RNA interference. Profoundly affected all mammalian virologists. In fact, most people who studied anything to do with mammals, this was a big deal, because now you could use a machine to make a small RNA to knock down any gene of interest. So we just never had genetics in mammalian systems. Like, you could make a knockout mouse. You couldn't make a knockout human. Knockout mouse took years, to really do it well. So this revolutionized things on many levels. Technology-wise, this was a revolution. Basic biology-wise, though, is where I got interested. So we and many others, my own interest as a postdoc was, so RNAi exists because double-stranded RNA, as you guys might know, is something foreign to our cells. So if you want to defend against pathogens, you want to attack those cells or those pathogens, but only those cells that are infected with those pathogens, not your own cells. If you attack your own cells, you get autoimmune disease. And so what chemically is different about virus or other pathogens? Well, double-stranded RNA was the answer. And so in plants, insects, nematodes, and lots of metazoans, lots of non-mammalian, non-vertebrate metazoans, RNAi is the antiviral defense. Double-stranded RNA is recognized by the host cells amplify it, and then they use those snippets of RNA to attack the virus. And we all wanted that to be true for mammals, and we've been studying this for 10 years. It doesn't seem to be true in mammals. If anything, mammals have inverted the process. They're using it to silence their own genes. It's to their own advantage, and if anything, they turn down RNAi when they sense a virus instead of turning it up. But we were all trying. All of us virologists wanted to study the hot thing, which is supposedly an antiviral response, and we all have failed. And then there was a big series of papers that came out that there was this other thing, which I had never heard of at the time. This was 2002, called microRNAs. And microRNAs are these small RNAs that once RNAi evolved to combat pathogens via double-stranded RNA, microRNAs are inverted hairpins of our own genes that basically host cells turn this machinery around to fine-tune gene expression. And it was very exciting. So all of us that have ever done northern blots look at RNA levels, we would throw off the bottom of the gel, and you'd see intense little lithium bromide staining nucleic acid at the bottom of the gel. We'd think it was just degraded crap. But it turns out that's where this amazing biology was. And so these RNAs, whether they're RNA interference, canonical by a virus, or our own cells using it to regulate genes, they make about a 20 nucleotide RNA that all of us were discarding, was throwing off the gel, and there was so much biology in it. And guess what? It's tied to the RNA interference gene. So I saw this as another chance to maybe study RNAi in viruses. I was training to become a herpes virologist, a type of herpes virus that causes tumors, and I thought, and dumbass luck, I thought correctly that if any virus is going to make these microRNAs, it would be the bigger the virus, the better. And I was studying a big one. Remember, that's why I went there, to have more open reading frames. And so I got lucky. The viruses I was interested in made these microRNAs, and we were early to appreciate, one of the first to appreciate that they would make them. We were one of the first to publish they made them. We were one of the first to figure out what they did. And from there, we kind of turned that on its head, and now we study. We're one of the better labs in the world at finding these things made by viruses, and we and a lot of others now are trying to figure out what they do, and then also how the host uses microRNAs to regulate the immune response, virus defenses. And from that, there's been like all kinds of new classes of RNAs that have emerged since the discovery of RNA. There's like a Pandora's box of new biology lying there that the technology just didn't even exist to find. So RNA has been amazingly exciting to this first field I've lived through. It's changed us for technology. There's a beauty to the biology. There's a beauty that we were throwing it out. There's a dumb luck story. And if you don't care about all that, it's going to lead to new clinical therapies, and it's now a multi-billion-dollar industry, all from dumb luck and honesty. And so it's exciting. And you know, to any of you that go on to science, to me, this is a story that what else are we throwing out, or we don't yet even have the technology? It's kind of like it was right in front of our face and we didn't even see it. So that's what microRNAs are. These are small RNAs that fine-tune gene expression. Now they suck to study in some sense because you want on-off. You want to study something, you better damn well sure it's doing something. Well, in microRNA world, 50% modulation of a gene expression is like, that's intense. Whereas in the traditional world, you want 50-fold regulation. It's easier to study the larger the dynamic range. So these are maddeningly subtle, fine-tuned regulators that play enormously important roles in biology. Anything that requires homeostasis, temperature, or, hey, the immune response. Too much immune response, bad. Not enough immune response, bad. So whenever transcription's involved and you don't want to have too much transcription, when you want an eyeball to have mostly eyeball proteins but not skin proteins, this is where microRNAs play a role, post-transcriptional. Long-winded answer, but that's what they are.
Undergrad students who want to go into research or medicine?
Shit, yeah, I got lots of advice. Okay, so this will contradict what I was saying earlier. So earlier I was saying that I don't understand how young people know what they want to do unless their parents did a number on them and beat it into them. With that said, it's such a competitive world that if there's a chance you might want to do it, you got to get involved early. And whether it's medicine or some other professional school, including scientific research, you need letters of rec and experience that matter because you're competing against people that are going to have it. So my advice, my strongest advice, is find a lab to do work in, meaningful work. It won't get you a nickel, and it's competitive to even get that free position. You will compete against 10 people to get one spot in my lab, maybe 15 people. I torture you if you're one of the 15 I select and make you come for 6 months just to hang out at my lab meeting before a spot even opens. But it's worth it to you guys. If you're lucky, you'll have what happened to me only when I was in grad school, and you'll fall in love with it. You would do it even if people didn't pay you. But if not, you might learn this is not for you, and a research track graduate profession is not. Or maybe you want to be an MD, you don't want to do research, but understanding the trenches of biomedically relevant research will help you be a better physician. And having something meaningful to write in your essays, statement of purpose, something real world besides shadowing a doctor because your uncle, was a doctor and could get you in, and you were lucky enough to get somewhere local you could shadow. I think that's a matter. So to try to avoid being as long-winded as before, get into a lab, be persistent, find a way, even if it's only for a semester or two. See if you like it, learn what the trenches are. Maybe you'll love it, but you will not get into a research track graduate program anymore unless you have research experience. Frankly, we don't want to take someone who's a senior. The younger we can get you, assuming you're not just doing what you're supposed to be doing apparently, the younger we can get you, it takes about a year or two to get you trained where you can become somewhat independent and start giving back. So we need you at least 2 years to get anything in return. It's not a selfless endeavor on our part. We want the free labor and make some discoveries together and also enjoy your company. You want a letter of rec that's meaningful and maybe get some experience. So beat down the doors to get some real world experience. Now, if you know hell or high water you don't have any interest in research, then do the equivalent. So if you're going to be an MD, find out what it means to be an MD before you go to that. Or a dentist. I never understood dentists, to be honest. Incredibly smart people, but a very high suicide rate. Think about it. This is all the rest of your life. Think about it. That's my suggestion. So when I was an independent agent, before I ran a lab with youngins under me. The highs were punctuated moments of glorious high where you knew something no one else did, where you discovered something, sometimes through luck, sometimes through perseverance. The 99 times you bang your head into that fucking wall, that one time it knocks down. It never knocks down, but it does 1 out of 100. And so the type of person attracted to research is cued by a lot of reward over a very short period of time, mixed with 99% of the time of misery and pain. And so if that appeals to you, if you're a gambler, it's not for everyone. That's what it was. So I can remember moments in the darkroom back before we had computer technology do it, where you look at an autorad film to see if a particular microRNA or protein's there, and 99 out of 100 times it was depressing. You want to cry, like someone punched you in the belly. But that one time, it works, and you knew, shit, I'm going to get a faculty job. You knew it. Like, I have the world's first viral microRNA. I ended up not being the first to publish it, but we knew we had it. Those high moments in the selfish years. And now it's the same thing, but when my people do it and seeing it through them and taking credit in the pyramid scan that is science for their brilliant young minds, and my old mushy mind can't come up with any ideas. No. So it's about the same thing. And then there's more paternal things now, like seeing them get where they need to go career-wise. Those are the highs. The lows are many, unfortunately. It's trite, but there's an amazing funding crisis for scientific research. We have academics are culpable for this, as is the government. The government's been a stagnant funding period. And so in inflation normalized dollars, we're about funding at the level we were 15, 20 years ago. That's backwards. So if you want the US to be a world leader in biomedical research or any of the sciences, that's back-asswards, and I have a real problem with the government on that. On the other hand, many of us have been sucking off the teat of government, including UT now with the med school, although I'm so glad I came here. It's going to be helpful for our research. But many places see the federal government as a gravy train to pay for large chunks of the salary of their faculty. So not so much the Texas schools. Texas schools, 50% of the salaries of medical schools are paid for by NIH dollars. At the university here, only 25%, but at some places it's 100%. That's just the system. Even the NIH will even pay for the buildings. So over the years, we've been adding more and more researchers competing for less and less, inflation-adjusted dollars. And so the frustrating thing is we could do more work if I wasn't writing grants, begging all the time. That's one of them. The others are where I once thought I was bulletproof and invincible at picking people to work for me that are better than me. I've made some real stinky decisions occasionally that I'd never made until, you're in the trend and you just make those. And so it kind of sucks. It kind of sucks to put your team at risk by bringing in one bad apple. That happens. So those are the lows. More than made up for in all the highs.